In this video, I'm going to show you how to use the Fiji macro to quantify the number of droplets per cell. So in the previous video, I showed you how to download Fiji and install the required plugins for this macro. So now I'm going to open again Fiji in here. And then I'm going to open the macro. So it's very easy to open the macro. You can just drag and drop the macro into Fiji. And it opens this file. Okay, so this is all the code that is run when you run the macro. You don't need to understand what's in there. You just need to run it. So you can either hit Control R or go to run here. If you click in here, you see the run button. So you can run this button, hit this button, and then you have the different parameters that are required to run the macro. So first of all, the input directory is where you have the images. So maybe we can start with this. I have two images in here. So I have input folder with two images. And these images show two channels. So in red we have BodyPie and in blue we have DAPI. And then I also have the same images just with the BodyPie channel to show you that you can also run the macro if you only have BodyPie channel. Okay, so if I run again the macro, I need to tell the macro which folder to use to get the images. Okay, so I'm going to start with that, browse, and I'm going to take the input channel with input folder with both channels, so DAPI and BodyPie. So in that case, I need to tell the macro which channel corresponds to the droplet channel to the BodyPie. So here it's the first channel and the nucleus channel is the third one. So using DAPI might help for the cell segmentation. We use CellPose here, very powerful cell segmentation, and CellPose was trained to identify cells with input images with one channel with cytoplasmic membrane marker basically, or with two channels adding a nucleus marker. So then associated to CellPose you have two parameters. First one is the cell size, which corresponds roughly to the diameter of cells in pixels. So here with this resolution I know that the cells have about 65 is a parameter that works well. If you have a lower resolution, you can reduce this parameter and you will have a better segmentation. If you have higher resolution, you need to increase this parameter. And then you have a threshold for CellPose. So in CellPose, the threshold is not exactly corresponding to a probability. And this parameter can be set between -8 and +8. Ideally, 0 works well. Okay, so in that case we're going to use 0. I know that it's going to work pretty fine. If you find that you don't segment enough cells, you can try to push it by having negative values. In that case, so it's going to help. Main caveat when you do that is that generally you overestimate the area of the cells. So depending on your biological objective, it might be a problem. For example, here you might find droplets outside the cells that you consider in the cell. And so depending on what you do, might be a problem, I don't know. If you find that you segment too many cells, you can increase this parameter with a positive value. Hopefully 0 will work fine. And then we also have parameters that we can use to filter out cells that would be too small. So here, cells that have an area inferior to 1000 pixels are just filtered out. So this, again, depends on the resolution of your images. If you find that you actually identify segment cells that are really small, then you can increase this parameter. If you miss cells, especially if you have large cells but you miss small cells, you need to decrease this parameter. Second set of parameters are associated to the Laplacian of Gaussian. So here we use the Laplacian of Gaussian as implemented in TrackMate. Very efficient implementation of the Laplacian of Gaussian. And we have two different parameters. The first one is the radius for droplets, so it's the scale of your droplets. So when you use the Laplacian of Gaussian, you're going to segment objects that all have about the same size. And so here, the assumption is that you want to identify droplets that have the same size. So you need to use a parameter that is well suited to your droplets. And you'll see in the results, so there's a result image that is generated by the macro that show you droplets that are segmented, overlaid on top of your image. And so the size of the circles, we're going to inform you about the size of the objects that are segmented. So if you find that your droplets are larger, you should increase this value. If you find that they are smaller than the circles, you should decrease this value. And you can have decimal values here. And finally, you have the quality, what is called quality in TrackMate. So it's a threshold. So basically, if you increase this value, you're going to have more droplets. If you decrease this value, you're going to have less droplets. So if you think that you are under detecting your droplets, you should decrease it. If you think that you're over, that you are finding droplets that are actually part of the background, you should increase this parameter. And then just be careful if you change the size, the value of the quality, so as it depends on the size, it's not going to be the same. So for example, a threshold that would work fine for a given size won't be the same for another size. So finally, you need to define where you want to put your results. So here, I'm going to create a new folder. I'm going to call it output. And the file suffix is the suffix of your images. So if I go back to my input image, I have TIFF images, okay? So I need to specify TIFF. The advantage is that if you have other kind of files, like CSV files, TXT files, they won't be taken into account. We'll only consider TIFF files. So if you use constructors or images out of a microscope, like CZI, you should put CZI in here, okay? And so then when everything is set up, you can just hit OK. So here it's not it's not run as in the background. So it opens the image, everything is showing up. So as long as you have things that are going on, you know that it's processing. So here's the second image is being processed. So first step is cell post, take a little bit of time, and then everything else is processed. And now it's down, you see, nothing is happening, everything was called. So when you don't have any image open, you know that it's over. And then you can go to output. And so here you see for each image there's a result image, a CSV file, and there's also a parameter CSV file that is generated. So here I'm going to show you what it is to start with. So this is to save the parameter that were used for this processing. It's really important so that you know which parameters were used, because you know whenever you process data you might not be sure about the parameters that you used a month ago, a year ago. And here, at least as the results are generated in one folder, as this file is generated in this folder, you know that the results you got, they were generated with this parameters. And here, so it just recapitulates. Droplet channel is first channel, third channel for nucleus channel. So here we see that we used 2 channels to do the processing. Cell size for cell post, threshold, minimum cell size, and so on. Log radius for droplets and log quality for droplets. Okay, very important. We are in the fair era, and so we need to do reproducible processing. The result images look like this. So here is the first image. Let me put it full screen. So it's the image. We see the original image with both channels, blue and red. The cells are outlined in yellow, large structures, and then The droplets are also outlined in yellow. So I told you droplets, we use the log, so it's related to a given size, and here you see the size of the droplets. It looks like the small radius. So you see that if you think that this is not the right scale for your droplets, you could increase or decrease the size. So here it looks fine. As you can see, we might have droplets outside the cells. They won't be taken into account, and also all the cells that are on the border of the image are not taken into account, and that's because if they're on the border of the image, they are only partially visualized, partially observed, and so then the number of droplets for a partial cell wouldn't make sense, so that's why we get rid of them. Okay, and so we have the results for the first and the second image, and if I go back to the folder, I have a corresponding CSV file, so I can open it. And here, as you can see, you have the cell ID, the number of droplets, and the cell area. So as the main objective here is to count the number of droplets, this is really important. And as we have the ID, so if I go back here, you see you have a number inside the cells. For example, here we see we have cell number 26, and we see we have 3 droplets. So we can here, so at least the result image, you can go back to your image to make sure that it's doing the right thing. So if I go to 26, I actually have 3 droplets. So you see you have correspondence between your image results and your file. So then you can do some stats with this file. And you have, so I'm not going to show you, but you have the same file. We can open it. It's going to be very similar here, except we have less cells, only 29 cells for this image, but quite some droplets. If we look at it here, see you have 22, 19, 21, 5 that are full of droplets. And if I go back to my file, I can see that I have 97 droplets for cell number 10, 147 for number 19, and so on. Okay, so that does what we wanted the macro to do. I'm just going to show you that you can also apply the same macro if you only have one channel tagged with BodyPy. So if I go in this folder, you see you have only the BodyPy channel. So let's run again the macro. I'm going to change the input directory here. So I'm going to select input BodyPy only. So I can use, more or less the same parameters, at least the ones that are related to the log, because it's been done just on the BodyPy channel. And for cell pose, I'm going to have to say that nucleus channel now isn't there. So I'm going to put -1. So 1 for droplet channel, -1 for nucleus channel. And I'm going to put the results in a different folder, output BodyPy only. Okay, and I can run it, and so you see it opens the first image. So here we only have one channel now. Processing, cell pose, log, extracting the results, going to the next image. Cell pose is processing. All right, everything is now over. And so if I look at the result, so you see it's very similar to before, but we don't have the DAPI channel. And quite amazingly, the cells are still quite well segmented, even though we don't have a DAPI channel. So that's quite useful if you only have BodyPy channel, for example.